Okay, so now we're going to look at the same parent genotype of big T, big R, little r. And we know that the gene for tallness has two alleles, big T, little t, and the gene for P shape, round or wrinkled, is going to have two alleles. And we know that they're on different genes. But let's just take a look at what the meiosis would look like if they were on the same gene. So, but they're not. But again, I want you to understand the concept. So just want to show you how this would look different. So if we had a cell that was before meiosis and needed to just kind of show we have I'll just show a different color here. We have one chromosome, and again, we have two copies because the pea plant has a biological mother and a biological father. So I'll put the recessive versions on that one, and I'll put the dominant versions on that one. And again, the reason I'm putting the dominant versions on just one and not having it be mixed is because I'm assuming that this was the result of a parent cross where we had all dominant times all recessive. So the little t and the little r would have been inherited together, and the big T and the big R would have been inherited together, right? So just kind of, I just want to show that it's possible that it would be different, but I'm just showing it this way. So we need to duplicate the DNA in S phase of interphase. So we're going to show that quick. The chromosomes again are going to look like X's now because there's two copies of each from each parent. And of course, when these separate and go through meiosis 1 and 2, we're going to end up with meiosis 1. Here we go. And then in meiosis 2, these are going to get pulled apart even further. And we're going to end up with 4 reproductive cells. And I'm not even showing the chromosome. I'm just showing the trait. So notice here, this is different because there's no independent assortment at this stage. There's just one chromosome we're talking about. So the only combinations we're going to get is big T, big R, and little t, little r. So if we were going to, what gametes are we going to get from that? We're only going to get gametes that have the dominant or the recessive trait. So we would only be able to pass those on together. So that's why they're called linked, that we're not actually seeing any heterozygous. We're not seeing like big T, little t, or we're not seeing those together. We're not seeing the big T, little r together, for instance. Right, we're not seeing that one, or we're not seeing the little t, big R. We don't see those. So wait a minute, what about crossing over? So at this point in meiosis, when the chromosomes are next to each other, we sometimes get, we get these things to like kind of cross over each other and then split and attach on the other side. So because of crossing over, we will be able to produce a small amount of gametes that have those hybrid combinations. Okay, so again, just kind of showing that a little closer, showing that step a little closer here. When we are, I'll kind of show those here. When we have that chromosome, it's the same chromosome, but it's the other parent. Okay, and if we have these two moving across each other, right, then the result of that is going to be those are going to be normal up on the top section. But then the bottom section here, we will have a section that has crossed over and is now different. So this is going to go there. And these two have now moved across, and you're going to now be able to produce some of those hybrid genotypes. Another idea is that crossing over happens more frequently if the genes are farther apart from each other. So like if you have the T up here and the R down here, there's a lot of space between them, and so it's more likely that they'll cross over and you'll get combinations that are different. If you have like the T here and the R here, the chance of making a break like right at that spot is less likely, right? There's more spots for breaking the chromosome if they're farther apart. So you get crossing over more frequently if the distance on the gene, the distance on the chromosome is greater. It's more frequent, and it's less frequency if the genes are close to each other. So this question is about that concept. Okay, so let's say we have a chromosome. I'm just going to draw the chromosome horizontally here. And on this chromosome, we have 3 genes, gene A, B, and C. And we want to know, where are they? Is it like this? Is it like this? Oops, that's the same thing. Okay, how are they positioned? How are they oriented relative to each other on this chromosome? So let's look at the frequency of crossing over. A and B cross over not very frequently at all. So because they don't cross over very frequently, they're very close to each other. And A and C cross over a lot. So that means that they're very far from each other. And this is kind of the in between, right? That's also, they're pretty close to each other. But so what I'm going to do here is I'm going to say, well, A and C are very far from each other. They're farther apart than anything else. So I'll just say A and C have to be far away from each other. So where do I put B? Do I put it here? Do I put it in the middle? Do I put it closer here? Well, let's see. What is B closest to? Well, B and C and A and B. So we have a 10% crossing over versus a 5% crossing over. So B and C cross over more frequently than A and B. So that means B and C have to be farther from each other. So B is a little closer. So A and B cross over 5% of the time. B and C cross over 10% of the time. And A and C cross over 40% of the time. I know 5 and 10% added up doesn't equal 15. And I just wanted to get you the idea. I just totally made up this question. So I probably could have written it better. But it just kind of shows the concept that you want to place the ones that cross over the more frequently are farther from each other, and the ones that cross over less frequently are closer to each other. Okay, I'll pause the video and then we will look at some more questions on crossing over and then a couple other concepts like incomplete and